cellular monitoring the structural cellular structure like actin and laminin specific chromobodies. Some labs have also made nano boosters. They are nanobodies directly again endogenous the proteins which are expressed endogenous GFP and RFP. They are ideal for super resolution microscopy or traditional fluorescence microscopy. Apart from these imaging, they are very good source of co-crystallizations as a co-crystallization chaperones. If you are in the crystallization fields, there are many proteins, challenging proteins where crystallization is tedious or not possible. With the help of nanobody, these non-crystalline proteins can be crystallized. Here are a few examples. With the nucleopore complex and nanobody, which was alone a nucleopore complex of 62, 58 and 54 was not able to crystallize. But once we had the nanobody which is binding to this complex, the protein was crystallizable, and we could determine the structure. Nanobodies are also used for intracellular imaging of nuclear pore proteins. Here are a few examples, which are, if you see the comparison to conventional antibodies, they are very specific, and you can see very clean images. Now for the applications, single domain antibodies are used for medical imaging. Researchers in many labs have used radio-labeled nanobodies for imaging the tumors. Like in the above picture, you see that the animal implanted with pancreatic cancer could be imaged. The tumor can be imaged using class 2 MHC nanobodies. The myeloma tumor can also be detected using anti-CD11B nanobodies, and there in the last picture on the upper panel, you can see that animal was imaged for the tumor with the help of PD-1 antibody. These are different cancer markers which are used for imaging different type of tumor and cancer. Rheumatoid arthritis can also be imaged using anti-macrophage receptor molecule nanobody. Many cancer markers are used. Nanobodies are developed again many cancer markers, and in many labs using a mouse model or any other animal model, they can show that cancer can be detected. In the upper market, the prostate cancer, which can be detected using PSMA VHH antibodies. Using CD38 VHH and nanobodies, multiple myeloma tumor can be detected. Using HER2 and nanobody, the breast cancer can be imaged. Nanobodies are also developed against many checkpoint markers. Here is one example of PDL1, where the PDL1 can, with the help of nanobody against PDL1, the cancer development can be detected, which is good for when the patient is treated with the cancer therapy, to see how good the patient is behaving with the therapy. Apart from the visualization, the many labs have developed many nanobodies against various toxins. These toxins are some are foodborne toxins like alpha toxin and mycotoxins. In the right hand side, you will see that researchers have lab have developed nanobodies against mycotoxin, and they can detect mycotoxin in various food contamination. They have also developed, some labs have developed alpha toxin nanobodies. In the left hand side, there are many nanobodies developed against various toxins like scorpions and snake venom, tetanus toxin, and some poison chemicals like resin from resin and clostridium toxin, which are bacterial pathogens causing various diseases, gastro or food poisoning. These nanobodies can be used for detection of various toxin, which can be used either in as a point of care or contaminations. There are the few examples where pathogen can be detected, which is mostly affecting the respiratory or gastrointestinal infections like Streptococcus mutans, Helicobacter pylori infection. So, for all against all these bacteria which are causing these infections, the nanobodies were developed or discovered against surface antigen of these bacteria or viruses. Um, Apart from discovering how these nanobodies can be used for making the point of care device, so many labs are using this nanobodies in a point of care device on microfluidic device by utilizing gold nanoparticles and making either paper based diagnostic or microfluidic channels. Here is one example of where one lab have developed a lateral flow assay for rapid Zika virus detection. And in another example, they have made a portable high sensitivity detection of salmonella using a nanobody as a capture molecule. Since they are small in size, making nanobody in a various formats is very easy. They can be used as a multi-specific VHH by just plug and play by combining different nanobodies together with small spacers or other various tools. This is one example on the left hand side of the lower panel that one lab have used pentameric nanobodies. It means 5 nanobodies they have used as a capture agent for detecting salmonella. Um, in one lab, they are using a platform, what they call it nanoplex platform, which is mostly used for fluorescence microscopy. If these this nanoplex platform is good when you want to see a good and clean image using various signals from different sources. So various signals from these nanobodies are detected, and one signal is made, or the other noise signals, what we call, can be erased. And the platform is very versatile, because then in that case you don't need high end instruments in every lab. So you just need secondary antibodies to cancel the signals. And nanobodies are also target for the tumor environment. They can be used to design imaging probes for different imaging techniques like PET or SPECT imaging, MRI or CT scans. There are some nanobodies which can be used as a diagnostic. The same nanobody also can be engineered as a therapeutic to break immunosuppressive response, and this can be a good target for a theranostic tool where precision therapy is required. Uh, dev- uh, Various nanobody formats are used, and people are trying to make, and they have seen that nanobodies are quite stable in various format. Like they can be labeled with easily with the fluorescent molecule. They can be labeled with the radioactive molecules. They can be labeled with the gold nanoparticles. Two different nanobodies can be attached, two or three. Why? Because small nanobodies have very short life, because they pass from the kidney very fast. So if they want to use in therapy, you want to extend the lifetime in the body. So this their size can be increased by combining two different specific nanobodies against several targets. Nanobodies can be constructed or humanized using different FC region of conventional antibodies. They can be used as drug conjugates for delivering any drug into the patient system. Now, nanobodies are also used in C therapies. Here is the few example for therapeutic application. Many single domain antibodies are used are developed against therapies of various diseases like inflammatory autoimmune disease, cancer disease, bone disorders, neurology, hematology, infection, and diagnostic. In this table, you will see that in the third line, what they indicate for different disease it's mentioned, and what type of format is used for the clinical phase, and on the right hand side on which phase of the clinical trials these nanobodies are. Some case studies for therapeutic nanobodies. This is one example that where VHH clones were able to block the epitope of both LRP and LRP6 proteins, and they can see the Wnt signaling. Um, uh, Wnt signaling response in stem cells. Um, Another study where in COVID-19, the single domain antibodies were raised against the spike proteins, and many labs have developed nanobodies which can neutralize the effect of SARS carcinoma virus. Um, there is this one clinical example of single domain antibody, what we learned before from Abling's Caplacizumab. It is now approved from European Medical Association. This nanobody is used to treat acquired thrombic thrombocytopenia purpura. It has a very rare autoimmune disease where our body itself mistake our own protein as a foreign protein. So our immune system respond to the our protein itself, and with that blood clotting start happening in a small blood vessels, which is very dangerous. So they have done various studies, and with the I think they are now in clinical 2 trial. Another example is breast cancer imaging for diagnostic. Here they have used radio label anti-HER nanobodies and used gallium 68 for imaging the breast cancer in the patients, and with this and nanobodies both type of carcinoma can be detected, which is in a primary stage, in a very early stage, or in metastatic lesions. um, like conventional antibodies, single domain antibodies are now also uh, mm, used or they are trying to use as a drug delivery system because of their small size, and by decorating the drug with on the surface with many nanobodies. There is a few example in which formats the nanobodies are used for a delivering partner. We have seen few examples how single domain antibodies are making clinical impact. They have both characteristic of small molecules and conventional antibodies. Apart from that, they are easy. First of all, they are easy in manufacturing, very stable, so which lowers the cost. How single domain antibodies are discovered. So they are derived from camel family, but we don't have to sacrifice the animal. We just need few ml of blood from the camel or camelid family, alpaca or llama. and they can be selected via phage display technology, ribosome display, mRNA display, or microbial display, means either bacterial display or yeast display. But most of the time you will see the papers, or most of the time the easiest technique what is people or widely used is phage display technology. Now with the artificial intelligence coming too much in the our daily life, these are also revolutionizing the nanobody discovery. How they are doing it, by using the generative AI and large language models, we can make synthetic libraries instead of immunizing the animals. Since there are now a lot of data available, the synthetic library can be made in silico, not made but not synthesized but plant in silico, and then using synthetic biology they can be made. So for that, for any pathogenic, they are good when any, let's say, some lethal toxin, especially in the case of snake venom or poison, we don't have to immunize the animal because it could be lethal for animal as well. So using the AI, the synthetic nanobodies can eliminate the need of animals and reduce the reliance on existing libraries. With the synthetic library, with the synthetic approach, they also reduce the time, because now we have so much language, biological language models, where you can predict with the AlphaFold and any other models, protein production interaction can be made in silico, and several candidates can be selected after sequencing before going to the wet lab. Here is the few example where in last few years many papers have published where they have used a synthetic library and discovered many nanobodies against pathogenic bacteria or venom toxins. Now what we are how we are leveraging the nanobodies in our company. We are using the first of all, we are making single domain antibody library platform. The library platform, we at the present what we have is three. We have immune library, which is specific for snake venom. We also have a nave library, and we also making a synthetic library where we don't have to rely every time for immunizing and the animal. uh, so the first focus of our company is snake envenomation. A snake bite envenomation is a acute, life-threatening, time-limiting medical emergency. It is in the priority list of neglected tropical disease, and it is declared by WHO. And you will be amazed to see that India is known as a snake bite capital. More than 50% of snake bite death happens in India. Around 2.7, around 81,000 death happens globally, and in that, more than 50% is from India. The main problem of snake, the main snake bite happens with the big four. What we say is Naza Naza, cobra, Russell viper, crate, and saw-scaled viper. Uh, but in India, apart from these four big snakes, there are more than 60 venomous snakes, and against that, there is no anti-venom. With this snake bite, in most of the cases, if patient has not approached to the hospital, the death occurs, and even if the death is not there, there are lot of disability, and every year around 400,000 patients left with a permanent disability. This is the problem of most rural area. So if any patient or any family member is bitten or disabled by the snake bite, there is a huge economical burden on the family. In the snake bite innovation, the first 6 hours is the golden hour. So if the patient has not approached to the hospital from the snake bitten, the first 6 hours is very very important. If the anti venom is not given in this duration, or if the right and if the right administration is not happening, it is causing many side effects. The problem with the current antivenoms, these are horse-derived polyclonal antivenom, and they are raised again only big four, what I mentioned before: cobra, crate, Russell viper, and sword-scale viper. Which it means they are not effective apart from these four snakes, especially in northeast where Naza kauthia, pit viper, in south part of India also. These antivenoms are not very effective, and especially in the case of pit viper, they don't work. And apart from, if the doctor doesn't know which kind of snake has bitten and antivenom is given, they have a lot of dangerous side effects. So, for the right administration, what at present doctors have, there is no diagnostic kit available or no diagnostic available to tell what kind of snake has bitten, unless and until patient has seen the snake or they know the geographical location that okay, in that place certain kind of snakes are available. Um, so this animal based, and looking at the Indian geographical variation, this animal based discovery process is fundamentally broken for anti-venom development, because first of all, snake venom variation. In the snake venom, there is a lot of variation, not only with the in between the species like cobra and crate, within the species, depend on the geographical location, age, and size. For example, the cobra of UP or cobra of Rajasthan or cobra of Northeast, though they are cobra, naza, but their venom variation is too much. There is too much variation. So the anti-venom, which is could be working with the UP or Uttar Pradesh naza, it won't be effective for the Northeast naza. For making antivenoms, the process is that snake venom, or the pool of snake venom, is immunized in the horse, and and after they produce antibodies, the blood is taken, and from plasma the antivenom is produced. Since they are animal derived and dependent on animals, so batch to batch there is a variability, and for every anti-venom you have to be dependent on horse or other animal, and for every species making anti-venom is not possible or very cost effective. And since they are conventional antibodies, they are not very temperature sensitive, so cold chain storage is required, and there is not enough supply. So looking at this, there is a very cost effective and very cost demanding, and they lack stability. This model is not scalable for regional wise snake venom anti venom preparation. The other focus what we have in our company is early detection of iron deficiency anemia. If you know about anemia in hospital, first of all, doctor do the hemoglobin test or ferritin test, but these are very late. These are not very accurate for early detection of anemia, and for this, there are in India is a lot of burden of IDA deficiency, which causes preterm birth, low weight birth body, low birth weight of a baby, and chronic fatigue. There are a lot of in rural areas miscarriages happen because of iron deficiency. How we are solving this problem? We are bridging this gap by using single domain antibodies. We are making the platform of single domain library using AI tools and immune library as well. We are developing nanobodies against various snake venoms, and we are designing their single domain antibodies for their diagnostic as well as therapeutic purpose. Um, How we do it, first we need antigen. In our case it's a venom or serum soluble transferrin receptor. We make a library. It could be immune library, phage or naive library or synthetic library. Once the library is ready, we do the panning against various antigens, and the panning is very stringent. We do various harsh conditions, and to have very specific, there is a lot of cross reactive biopanning and washing happens. Once we have the target nanobodies, we produce in E. coli, and then it goes to the development phase where we make nanobodies, or we develop biosensor using gold nanoparticles, and we make detection kits. We are in the development phase. So with our single domain antibody platform, we can solve, we are on the um, um, we are focused on solving the variation and regionability problem of snake venom. Our main focus is first of all the diagnostic, because there is no diagnostic kit available, not only in India, in the whole world. There is only one diagnostic kit available for snake venom in Australia, but this kit is will not work in India, as these are specific for Australian snakes. We can eliminate animal immunization. The process is entirely in vitro, and we are using E. coli, so it is very cost effective. How so for single domain antibody discovery and development, there are two different phases. First is discovery, and once we have the target molecule, it goes on the development part. For discovery part, we prepare antigen and immunize the animal. It could be alpaca. In India, alpaca is not possible, so we partner with other abroad companies. And in India, India is the big source, especially if we see the Rajasthan, we can use camels for immunization. Once the antigen is immunized in 6 weeks or 8 weeks interval in a small doses, which is not lethal, 100 ml of blood is collected from the animal, and VHH library is prepared in different, it could be, as we learned, it could be in different formats. We prefer phagmits, but it can be made in a ribosome, or it could be mRNA or bacterial or yeast library. Once the library is ready, the biopanning stage happens, where it's a stringent washing with a various condition, pH variability, cross reactivity checking, and we select various nanobody of interest. Normally, we get 5 to 10, from 10 to the power 8 library molecules, roughly 5 to 10 good candidates, which can be used for diagnostic and therapies, therapeutic target. Um, for development part, first, once we have the pool of, after biopanning, when we have a pool of nanobodies, we do the sequencing. This is de novo sequencing, because the sequences are not known. These are not based on any database, so they are a totally novel sequence. Once we have the sequence, we classify from the huge database which kind of classes of nanobodies we have. It could be based on the CDR, especially we focus on the CDR regions, because these are more target molecule for the antigen binding. So based on the CDR region variability, we classify different class of antibodies, and we select 2 or 3 from each class for rest process of the development. We do the single domain antibody affinity maturation. This can also be done via, so this is a mutagenic application. This can be done with the phage display or ribosome display. Once we have a very good targets, let's say 2 or 3 targets, we conjugate single domain antibodies. In our case, we use fluorophores or gold nanoparticles for calorimetric assay development to make a prototype. The single domain antibodies can be stability can be improved depending on so normally the nanobodies are stable up to 70 to 90 degrees centigrade, which is not possible with the conventional antibodies. They can be engineered with a bispecific or multispecific, means two different target candidates can be joined for making more high affinity nanobodies. Nanobodies can also be humanized for therapy purpose. So, as a conclusion, single domain antibodies are more than just a new antibody class. They are very versatile molecule. They are nanoscale platform that bridges the gap between biology, AI engineering, and engineering. They allow us to build the sophisticated medical tools what we need for future personalized and predictive diagnostic and therapeutics. At ProteinoMapTech, we are in a development phase of various target candidates. As a founder, I am there, and Dr. Sushmita is also the co-founder of ProteinoMapTech. We incorporated last year in 2024 March. We have received various government funds, and we are very proud for that. Presently, we are located at IIT campus in Bionist, and at present we have uh, several positions, and we always look for the students who want to do training or who want to do the training or do the internship. And we are also open for faculty collaboration, academic collaboration, if they have certain type of targets and they want to see or develop antibodies. So we do the collaborations. I'm open to questions now.
Hello, ma'am.
Participants, if you have any questions, please ask.
Yes, sir, I have a question. Am I audible?
Yes.
Ma'am, is there any marketed preparation with these antibodies for snake venom?
Pardon? Say it again.
Is there marketed preparation? Yes, ma'am.
Marketed what?
Marketed preparations like formulations, which is available in market
No. So
for general use.
classically, so basically, as I mentioned, for diagnostic purpose, there is no um, diagnostic based for either it is antibody or aptamer. Any type of diagnostic is not available for snake venom. The antivenoms are also the antibodies, but they are polyvalent. It means they are not specific for one target snake, what you say. And snake venom is not a single protein. So snake venom is a cocktail of hundreds of protein. So when antivenoms are produced, or they are produced, the whole venom is injected. So it's a pool of various target anti proteins venom. So there is not specific. So in India, there is only one antivenom at present, which is a cocktail of big four, means cobra, Russell viper, trait, and soy scale viper. And that snake venom is also from the south part of India. So they are antivenom specifically focused or not focused. The production is more focused, or the source of material or source of venom is south part of India. So this is one of the reason that antivenoms from especially in the northeast, they are not very effective.
Okay, ma'am. Thank you.
Good evening, ma'am.
Yeah. Good evening.
My question is that do we have any example of having the treatment using the single body antidomain in
As I
COVID-19?
In COVID-19, many labs, in our previous lab as well, in my previous lab, we have developed nanobodies against single domain antibodies. For diagnostic purpose, we were using it. But for therapy, there are a lot of regulatory process. So if you have seen the therapy table, so they have to, at present, not in the market, because lot of approvals are required. So there are few in clinical trials. for neutralization purpose. But a lot of nanobodies in labs, they have shown neutralization capability of SARS-CoV virus, coronavirus.
So a lot of clinical trials are running on, right?
Yes.
Okay.
Excuse me, ma'am.
Yes.
Thank you, ma'am. Thank you, ma'am.
Can you tell me some software tool for protein structures?
There are
Like?
many tools, but at present, if you see there as if you just want to know the protein structure which is not available in PTP or which is not solved in a wet lab by crystallization or NMR, AlphaFold is there. So if you put any sequence, you will get the structure of that protein. And if you want to see the sequence, the let's say protein-protein interaction or modeling you want to do, you can do the docking. There is a Rosetta server is there, and you can do Clustro is there. There are many servers. Rosetta VIP is very good.
Okay. Thank you, ma'am.
Hello.
Yes.
Yeah, ma'am, I have a one question.
Yes.
In these between the slides or in your presentation, you have used some microfluidics devices. I am working on that microfluidic devices, and I am right now I'm detecting the microRNA.
Okay.
So I want to know that which types of detection techniques will be the best suited for the detection of the microRNA. Either it's a electrochemical or the optical
It
devices we
depends
have to
what you want to see as an end product. If you want to see just the, let's say you want to detect whether it is present or not, it's quality or it is qualitative or it's quantitative. For qualitative purpose, calorimetric assay will be good. For quantitative, also calorimetric assay is good or will be in used, but you have to change the device, which could be not as a paper based, but the cuvette based.
But if the samples are the large, then which types of detection techniques are the most suited for the microfluidics?
Mm, in my personal, I will use calorimetric only, but electro nowadays people are using, and the reason is that if it totally depends if you have using proteins, how good they are behaving in that platform.
Okay. Thank you, ma'am.
In my personal, because personal opinion or what we are using or we would like to use is calorimetric, because in that case the small fluorophores can be attached easily, and they are very easy to attach in different let's say kind of N terminus of your protein or C terminus or protein, or you can make a system melamide labeling with a known target regions. But any type of microfluidic device will be good if it is for the point of care devices. The
Yeah.
sensitivity is one of the thing where you
Yeah.
have to.
And which types of fabrication techniques you have to use in the during your experiment in the for the microfluidics fabrication devices? Either it's additive or subtractive.
Uh, for that, I can't answer, because for this part, we are collaborating with the IIT faculties who
Okay.
is mostly from the chemical engineering field. So we are mostly for the biological part, and for the microfluidic, we have to collaborate. So we are
Oh,
collaborating. So of course, I can answer. I cannot answer right now, but I know your question. I will ask my collaborator,
no,
and
no, no, no.
you can
I just
contact us.
I
Yeah.
just asked because there are the different types of technologies have been used in the for the fabrication of the microfluidics. That's why I'm asking you.
Yeah. I am not expert on that. So if you want to ask anything, how you can make a nanobodies or how you can engineer in certain things, I will be the answer. But for the microfluidic part, honestly, this is not my expertise. I can tell you
Okay.
something what we are using at the moment and what we see as an example, which is working for us. But if you want to know the general what will be the better in your case, I think for the expert person will be better to answer. You can always write us and
Oh, okay. Thank you, ma'am.
Uh, Dr. Mamta, there is one question from the audience. Which imaging technique used for imaging nanobodies?
if it is only for the cell localization, protein localization, then confocal microscopy can be used. But if it is for the patient's PET or any high-end super resolution microscopy is also used. So I haven't seen any microscopy technology where nanobodies are not used at present.
Any other question, participants?
So I myself is working working on nanobodies from last 8 years, but in last 4 or 5 years, the papers what are coming in every week, it's really huge. It's really huge, and they are wonderful tools. And what I see that in India, not many people are using. So if you are interested, please do.
I think there is no questions from audience.
Thank
Thank
you.
you for taking time for us from your busy schedule. It's a
Thank
great
you.
honor for me. I request you sometime visit our college and also educate our students from the biotechnology and biomedical so that they can
Definitely.
have some
I
interest.
would like to use
And I
this
love to collaborate
opportunity.
with your company too.
Yeah. I would like to encourage students if they, I know Guwahati is very far from Rajasthan, but we are open to have intern students, and if you want to do any training, we are very open. Just write to us. We have contact number you can get from Dr. Jaiswal, and then okay. Have a nice evening, all. Yeah.
So participant, chalo. So I requested to all the participants kindly fill the form before the 7:20, and we will wait 10 minutes for the upcoming session. So the next session will be the resume from the 7:20.
हॉस्पिटल ये सब फोटो लानी है। वो बेटी प्लीज ये फोटो लानी है। इम्पोर्टेंट ले सकती है।
Hello,
Yes,
am I audible?
not audible.
Welcome, Dr. Manas. You are audible.
Audible, yes.
Yeah, am I audible?
Yes,
You are audible.
Some voice echoes are coming.
yes, your voice coming.
Hello. Welcome, Dr. Manas.
There is some echo.
Echo.
There is some echo at your end, I think.
Yes.
I think. Welcome, Dr. Manas. I think I am audible to you.
Yes.
Yes.
So shall we start?
Yes.
So I'm ready if it is from your side.
So, namaskar everyone. On the behalf of the Mody University of the Science and the Technology and the organizing committee, I welcome to you the next session, that is the session 8, day 4 of the ATAL-FDP program on AI and innovative engineering in the healthcare, from smart implant to the wearable tech. It is our immense pleasure to welcome Dr. Manas Thakur as accomplished intensivist and anesthesiologist and healthcare innovator to the ATAL-FDP program on the topic Innovations Research Solutions for the Patient Care. Dr. Manas currently serves as a chief consultant on the critical care and consultant anesthesiologist at the Sarthi Hospital, Assam, and is also co-founder and the director of the Medinnovation Research and Consultancy Private Limited, Guwahati, an organization dedicated to the bridging the gap between the clinical needs and the technology innovations. With his extensive expertise in the clinical care medicines gained during the high tenure and the prestigious institutions such as All India Institute of the Medical Science, Raipur, and the Northeastern Indira Gandhi Regional Institute of Healthcare and the Medical Science. Dr. Manas has consistently demonstrated the excellence in the clinical expertise, research, healthcare innovations. His insight on the integration of the artificial intelligence in the healthcare, especially in the area like smart monitoring system, precision medicines, and the biomedical innovations, will undoubtedly inspire the participant to explore the interdisciplinary approaches toward the patient-centered technologies. We are truly honored to have him with us and look forward his valuable contribution that will enrich our understanding how AI-driven innovation transforming modern healthcare from smart implants to the next generation wearable technologies. Once again, we extended a warm welcome to Dr. Manas and express the gratitude for the joining us in the intellectual journey and towards the shaping of the future of intelligent and the innovative healthcare system. Welcome, Dr. Manas. The session is handed over to
Start.
you.
Thank
Thank you for this kind introduction. So if I am audible, I would like to share my screen. Is my screen?
Yes, Dr.
Okay, so thank you for inviting me in this ATAL-FDP. And I'm very grateful to the organizers for giving me an opportunity to speak to this August gathering. Today, the broad topic was AI and innovative engineering in healthcare. And there were various thematics in that. Of that, I would like to take forward two thematics. That is, one is entrepreneurship and product development. And second is integration of AI for real-time monitoring of health status. So many of the participants might be faculty in different organizations, and many of them might be innovators and who are thinking of developing their ideas into products, taking forward their journey. So I'm very grateful that at one point of time when I was in an academic institution before. So at that time we had this idea, and that idea we took forward, and right now we are about to launch our product in the market. So I will share the needs and the necessity and the steps of entrepreneurship and product development. Vis-a-vis, I will give the example and our journey, what we have done. So I am Dr. Manas Borthakur. So slide is not okay. Is the screen visible still?
Dr. Manas, your screen is not visible. I think you have to reshare.
Is it visible now?
Yes, it is visible.
So our product is continuous expiratory airway resistance system. We call it Spiro Plus. So it was designed to improve lung function and healthcare outcomes in patients who are on tracheostomy. As an intensivist, I encounter a lot of people in ICU for whom we have to give mechanical ventilation for a long time, and we tend to make a hole in their windpipe so that they can breathe through that hole. So these patients, we have developed this device to improve the outcomes. So I will go step by step into the journey of entrepreneurship and product development. So in that first step, for any healthcare entrepreneur should be that they should first assess the need of some solution. Whether there is a gap in the market, there is a gap in the society, which currently no one is talking about, or which they are not solving, which there are no devices or no medicines to solve that gap. And that gap is a genuine gap. If we solve it, that gap will really help the end user, in this case, the patients. So in healthcare innovations, earlier till now, till very recently, the domain is that the dogma is that the devices, medical equipments are made by engineers in the laboratories. After that, they provide that instrument and equipment to the end users, and the end users have to learn, relearn, or adjust the things and use it. So this concept was also there in the Western countries, but some years back, some MIT researchers, they postulated the reverse of this pattern. They said that the requirement and the guidance for development of the devices in case of medical fields should come from the end users. The persons who are facing the practical challenges, the person who are going to use that equipment or device, they should come forward with the problem statement. They should come forward with the need. So there they have what they have done. They have done to identify the need, to identify the problems. The engineers have gone to hospitals, to academic institutions. They spent time, they did internships to identify problems. So this is a good approach. The end user has to identify the problem. He will give you the requirements, what is required, what problems are there, what pain points are there, what are the practical challenges in solving that problem. And then the engineering team and the healthcare professionals together, they will develop a solution, they will test it, they will do multiple iterations, they will test it and again change it, again they will test, retest, rechange, multiple iterations will go into it, and then finally the ultimate product will come. And that should be the actual approach. And slowly I'm very happy that in India also that approach has been coming up, and recently more and more healthcare professionals are moving into entrepreneurship in medical devices, and they are trying to solve the problem along with the engineering backup. So in our case also, precisely the same thing happened. So if you can see these pictures, the first is a patient having head and neck cancer who underwent this tracheostomy procedure, making a hole in the windpipe. Second one is an old patient underwent stroke or neuromuscular diseases, road traffic accidents, which are very common in India, large thyroid swellings because of which patient cannot breathe, so we have to make a hole in the trachea via the thyroid. And in ICU patients having severe pneumonias, COVID, and clinical critical illness, they also underwent this type of procedures. So in our case also, this happened as a practical challenge. At that time during COVID, I was in All India Institute of Medical Sciences, Raipur. There were severe crisis of ventilators. People were dying due to lack of ventilatory support. So, and we were also helpless. We could not accommodate. We had a lot of ICU beds, but still the patient rush was so high that we could not accommodate. We had to refuse people. So at that time, a few clinicians, we thought that we have to find out the reason of ICU bed occupancies, which are reversible, so that we can free up ICU beds. Then we found out that these people who are on tracheostomy, maybe due to cancer, due to road traffic accident, due to thyroid problems, due to strokes, they tend to stay in the ICU for long term. And when we did a in-depth analysis for the reasons for being in the ICU for a long time and occupying the ventilatory beds for a long time, one of the reasons was that these patients were more prone to pulmonary complications. Then when we dig deeper, we found out the reason of these patients having more pulmonary complications was that the way they are breathing was not physiological. Actually, when we breathe through the nose and mouth, our lung, it expands. But in these patients, our nose and mouth is bypassed. So while they breathe out, the lung get collapsed. So there we identified the practical problems. So the practical problems in this situation was there, that high out-of-pocket expenditure on health, which is very high in India. And every year around 60 million people are pushed into poverty due to excessive healthcare expenditure. These people stay long time in the ICU and the hospitals, and they block the critical care beds and ventilators. And during pandemics and emergencies, the needy people do not get access to ICU infrastructure. So we did a clinical trial in AIIMS Raipur, and we found out that if we connect few already available devices in the tracheostomy, then people lung function might improve. However, when we did our first experiments, those experiments led to certain high work of breathing and problems in the patients. So we stopped the clinical trial in between, and then we went ahead to modify the design of our device, and we dug deeper, and we developed another solution for them. So first step is assessment of the need, that there should be a genuine need which needs to be solved. And once you identify that need, you have to have a team. That team will deliver you the results. Because suppose if today I think that I will make a nuclear reactor in the air, that is just a thought, but that is not practically possible at present. So the need assessment should be practical, should be realistic, should be solvable, and it should have a team who is passionate to solve the problem, which is comprised of domain experts of different domains. Because in healthcare, to develop a solution for healthcare, you need doctors, you need engineers, you need designers, you need people who are familiar with the infection control processes. So you have to have a team who will have similar wavelengths and similar aspirations. Then only the team dynamics will work, and together all your expertise and all your energies will be synchronized, and your product development journey will go forward. So in our case, we were a team of doctors from AIMS and engineers from IIT and NIT. And we had people from the medical device industry as well in our team. So we co-founded the organization with 5 people, and we had advisors from IIT and AIMS and other reputed central government organizations. So we were lucky enough to have a good team of people from different expertises, and we moved forward with our passion to solve the problem. Because in this, if you are not passionate, you will have a lot of roadblocks. The main roadblock will be that your team, you have to be together. There will be confusions, there will be conflicts, and you have to navigate through them. Second is you will be having a lot of problems for funding. So that funding problem is also one of the biggest problem in healthcare innovation, because the process takes a lot of time, lot of resources, and lot of energy. And fortunately for us, at present in India, I'm very thankful to the present government that they are promoting healthcare innovations, and they are giving grants to ideas which are promising ideas. At the idea stage itself, they are giving grants. Because for healthcare innovation, initial stage is very difficult. Till we don't have a product in hand, the industry doesn't sponsor it, because there are regulatory issues and no one exactly knows whether your product will get regulatory clearance or your product will get approval to sell in the market. So initial stage, at the idea stage, at the prototype stage, at the product development stage, you need to have helping hand from government organizations, from non-government, from academic institutions. So in our case, we were lucky in that case that we got different grants from the government of India. So after that, once you have the need assessment, once you have a proper team, then you have to plunge into developing a solution. The developing a solution, as I told you, will be a continuous process. It's not that you will develop one product and it will be the best product having the best fit for the market. It will be a continuous journey. You make something, then you test it, you change it, you again innovate, and for that you have to have out of the box thinking. And in India, I am very sure that there are a lot of people who are having out of the box thinking, because we are very good at doing jugaad. So when you have this out of the box innovative thinking, it will lead to novelty of your product. And as you know, for any product to get a patent, there should be 3 steps. There should be novelty, that your product should be novel. There should not be similar idea or similar product anywhere in the world. Second is there should be inventive step. For example, if I develop a fan, whose blades are not straight, whose blades are curved, or there are some other, but it functions by working by rotating in a circular motion. So in that case, there might be novelty, but there will be no inventive step. So you will not get patent for that. For any solution to be patentable, there should be 3 steps. First is novelty. The invention should be novel. Second should be inventive step. You should have invented something which was not existent before. Third is the thing which you have invented should have industrial application. It should help and solve some real problem. Only these 3 criteria, when are met, you will get a patent. So your product should be robust. It should not be like, it should be a performing product. It's not that it's very, very brittle or it breaks down very easily. It has got a lot of machinery which needs very frequent maintenance and very frequent changes. So it has to be robust, it has to be practical, it has to be affordable, and the most important point, it should have an impact. Only products which have impacts are the ones which will lead to disruptive changes and will actually help the society. One example I would like to give, this is I have read somewhere about the founder of Apple, Steve Jobs, and when they were developing the first, the iPads, iPhone, like that, not iPads, the music devices, the first music devices when they were developing, he told the engineers to make it small. Then they tried very hard and they told that this is the smallest size we can have. But he still wanted it further smaller. So the story goes like that. What he did, he took the device and he put it in an aquarium, and a lot of bubbles came out. Then he told that there's so much air in this, that means it can be made smaller. So our design thinking should be in that way. Like when a person will touch and feel the device, it should be ergonomically such that it should be easier to use. It should be practical. It should be smaller, as small as it can be. So these are practical things which we have to take into consideration while developing the product. So what we did in our solution, we did a novel design. We made a solution so that there were all these pain points we identified during the clinical trial which we did. So in the first device, there were a lot of resistance to inspiratory flow. So patients were having difficulty in breathing. So what we designed a thing which will mimic the natural way of breathing in these patients. And as it was novel, so we had applied for patent and we got patent granted in India, South Africa, and few other countries like Brazil, Philippines, Europe, USA. The filing process is going on. Then after that, what we did, we took our product, then we discussed it with end users, intensivists, ENT surgeons, neurologists, pulmonologists. Then we took their feedback and we refined our product according to their feedbacks. The ultimate product which we got, it was preventing the lung collapse and improved lung function. It enabled the use of spirometer in these patients. Till now, there are no devices that enable spirometry in tracheostomized patients. Spirometry means that blowing the balls, you must have seen, most of you must have seen that 3 balls you have to blow with your mouth. So they can be used with mouth, but tracheostomized patients cannot use it. So our device they will be able to use incentive spirometry as well. And it will decrease complications and it will decrease healthcare costs by decreasing the length of stay in the ICUs. So now what we are doing, we are integrating artificial intelligence and machine learning into this. So in the next version of the device, what we are doing, we have put certain sensors, because as the patient will be breathing, our sensors will record the peak expiratory velocity and the forces and the pressures. And we have developed an algorithm and software that will give us the real-time data on the lung function of the patient, how his lung is behaving. Like initially, when the patient was using the device, what was the status of his lungs? And with subsequent use, how much improvement his lungs are getting. So that thing also we have included in our device now. Our next R&D which we are doing is integrating artificial intelligence, machine learning, and waveform analysis into the device, so that we'll have an algorithm for predictive analysis of the lung function. So this is how we have to adapt to the changing needs of the patient, changing needs of the thing. Initially, when we were working on the sensors, we were working on pressure-based sensors. Then once we perfected the algorithm for the pressure-based sensors, we came to know that there are still better principles of recording flow than pressure-based sensors. So subsequently, we developed ultrasonic sensors as well. So that is how the journey of product development changes. You have to develop one product and continuously you have to think of how to enhance the functionality, how to integrate newer technology, how to integrate newer concepts into your product design. So like that only you will have a great product at the end of the day. Once you are developing the product, simultaneously you have to see the market size. What is the size of the market? How big is your opportunity? Whether your innovation is a market creating innovation. If such type of innovation was not existent in the market, for the innovation which you have done, because of that, certain new customers will be using that device. Okay, so, and if it is a market creating innovation, you have to see what is the opportunity, how much opportunity, and your analysis of the market opportunity should be based on realistic expectations and aspirations. For example, the spirometer market, this is the analysis we have done for our product. So tracheostomies, actually the market, there are market research organizations which do market research for you, as it is not our domain. So we have outsourced it to certain professional market research organizations, and they have given us the data. So according to them, in 2023, around 1,44,000 tracheostomies were done in India. And incentive spirometry market in 2023 was around 18 million US dollars. This was Indian data. But we have to be realistic in our expectations. So we know at present with our infrastructure, we will not be able to cater to the entire India at one go. So what we did, we took out the data for East India. How many incentive spirometry units are sold in East India alone? So we found out that around 350,000 units are sold in East India. So that is called, in terms of business, that is called service obtainable market. So our service obtainable market was, we call it SOM, was 3,54,000. Then of this, what units we can attain. Okay. So the total market will be the total market of entire India. Entire India was around 26,48,000. Of that, East India was 3,54,000. Then that is addressable market. And of that, we have to be realistic. So we thought that we'll take 10% of that share. Whatever was there in East India, we'll target 10% of that share. So we took the market analysis and we found out that for us, for the sales, we have to make around 35,000 quantities for initial part. So your expectation should be realistic. Your expectations and aspirations should be matching with the practical market scenario. Because as a new player, we cannot think that we'll capture the entire East India market at one go. There will be other players, there will be threats, there is this thing called SWOT analysis. So these are all business things. You people might be knowing better than me, all these things. Those analysis you have to do. Then you have to find out what is your market, what is your opportunity, what is the size of your market. Then you have to utilize your resources. You have to take risks, utilizing your resources, and these things you need to tell your funding agencies also that this is the market, we are expecting this, we are expecting that. So they will see these numbers very much. So next is, you have developed the product, you understood the need, you developed the team, you developed the solution, your solution is working, and then you have to develop a business model. What best will work for you? You have to develop that. So here again, I would like to give the example of Apple. When a person goes to an Apple store in the US, so whenever it is said that whenever a person goes to the Apple store, there are maximum chances that that person will buy an Apple product, or he will come back and buy an Apple product later. So their business model is based on solving the problems and the aspiration of the client. So their salespersons and their representatives are taught in a way that they have to go the extra mile to understand the particular need of the patient, customer, and to make them in a comfortable position so that they can buy. In India, when Apple came, initially the product price was so high, people were not able to afford it. Even if they like it, they are not able to afford it. So they innovated and they brought the no-cost EMI model. Now you all might be knowing, most of the Apple phones in India are bought in EMI. The people who were not able to buy the Apple phone earlier were now able to buy the Apple phone. And that is called the changing your policies according to the market dynamics. Second point is, your business model should be that you have to stick to your core competencies. You should not divulge into a lot of things. Here what happened, I would like to give you an example of the, most of you must be knowing, there is a puzzle company called Lego. The small parts the kids put and they make puzzles. Lego was a huge success, huge company at one time. Then they were having so much revenue and they were having so much surplus capital. What they did, they thought that we will diversify. Then they diversified their business into a lot of things. Lego amusement parks, Lego food, Lego this, Lego that. And what happened? Slowly they shifted out from their core competences. Their product innovations decreased, their new products were not coming, and slowly, the company's shares fell down and it reached a point where it was about to close. Then they understood their problem, new management came in. Then they again focused in their core competencies, what they did best. So they did best was making puzzles. So then again came it back to that. They studied their customer profile. The kids, even big adults also use Lego. So like that, they innovate their product, and now again they are a very successful company. So these things you have to know the story of different companies. I'm not saying that you have to know only about the successful companies. You have to know about the failures as well. Then only you will understand. Like you should know about Nokia, you should know about BlackBerry, what happened, why did they fail. You should know about SpiceJet, you should know about Jet Airways. So if you are trying to develop and go into a product development, so these are things which you should keep in mind. The most important thing is you have to understand your customers in and out. You have to know everything about them so that you can give them the best product. So this type of, you have to develop a business model. So when you develop a business model, you have to find out who will be your key partners, because all the things you cannot do on your own. So you have to take help of certain people. Like for our product, we need help of regulatory, we need contract manufacturers, we need distribution and marketing. Second is what model, key activities you will do. What are the key resources you will have? For us, our key resources, our R&D team, our patents, our regulatory consultants. So with this model, what is your value proposition? What problems you are solving for your end users? Who are your customer segments? And what are your revenue streams? What type of revenue generation you are planning to do? Like you will manufacture it directly and sell, or you'll collaborate it already existing players, or you license it to different players which will not be linked to geographies. So these are various revenue streams you have to understand. So you have to understand the business model. This is a short business model I am showing you. Actually, there is a business model canvas. There are a lot many other things are there, which you have to prepare for your product. And the better you understand your business model canvas, the more are the chances that you will succeed in your venture. So you have to judge the competition. When you enter the market, there is a thing called GTM, go to market strategy. So you have to strategize how you will enter the market. Who are your competition? Who are the players? How big are the players? Okay. Today, suppose I develop a solution, an operating system for a laptop, and I go directly and I am competing with Microsoft. So very next day, either they will buy me out, or they will decrease our sales, they will give their promotions and all, and will be destroyed. So we have to understand how big are our competitors. If they are very, very big, you have to try to understand their collaborative stance for your product. You have to approach them so that with their expertise, you can benefit as a separate identity. You have to see what is your edge, what is your USP for your product, what are your price points. So all these things you have to understand. So for us, we did the competition analysis, what were there, what was the price points, and how features, pros and cons of those products. So those researches you have to do. And lastly, you should have a roadmap, what you will do, in which stage of the product you will do, and how much funding you will need for your future. At any point of time for next 18 months, you should have your plan and your funds ready. Otherwise, in startup, there is a concept called the value of death. So in that part, most of the startups, they go down and they vanish from the market. So these things you should be very clear. And so this is the last slide. This is our journey. The left side top corner, which you are seeing, is the first product which we made. Then slowly we did different iterations. We did laboratory and computer simulation studies. And after a lot of iterations, we have reached to the present version of the product. So that is from my end. Thank you. Any questions and discussions I would like to take, I'll be happy to answer your queries.
Thank you, Dr. Manas. Participants, if you have any question, so please ask. Either you can write in chat box or you can directly ask. So, Dr. Manas, we have some questions from the participants. This is from Dr. Rameswar Sugandhi. And he's asking, what innovation can overcome spirometer techniques related error ensuring accurate lung function assessment for diverse patient populations?
Yeah, as I already told you that there are different technologies based on measurement of flow. So earlier in India, at present, most of the spirometers are turbine based. So they use a turbine sort of mechanism. It is a simple technology. So they are cheaper, and the problem with that turbine based solutions is that they tend to, the secretions which come out from the patient's mouth and all, they might get stuck there and decrease the functionality. And one important marker of the lung function is peak expiratory flow rate and certain forced expiratory volumes. So these cannot be given by the turbine spirometers. So for that, and the accuracy of the turbine spirometers are comparatively lesser. So now the technology which is coming is the ultrasound based spirometers, and one more technology it is coming is the oscillometry based spirometers. So they are more accurate. They can detect very, very less flow, and they give real time readings. I hope this answers.
Right. There is one more question. How can a spirometer be designed for better infection control and hygiene, reduce cross-contamination risk between patients?
Yes,
Hello.
actually the spirometers, they come with a
Hello.
I think someone is speaking. Okay, so the spirometers, they come with a bacterial filter, so that one spirometer is used by a single patient multiple times, and once it is used by the patient, the home spirometry devices, one patient will use once. The commercially available spirometers, they have antibacterial filters. So those antibacterial filters are consumables. So you have to change for every patient. So that way it protects you from the secretions and the cross contamination.
Right, Dr. Manas. There's one more question. What role do you think for portable low-cost spirometer in expanding lung function testing, assess in resource-constrained setting and the rural areas?
Yes, good question. The spirometers which are presently in the market are the ultrasonic spirometers are very costly. around 75,000, 60,000.
इसमें हरियाणा के 14 जिले फरीदाबाद, गुरुग्राम।
Yeah, so what I was telling that the spirometers which are ultrasonic developed right now are very costly, 60 to 75,000, which are not for our rural populace. So what we are designing are spirometers which will be both diagnostic and therapeutic, and it will come at around 1/5 of the cost, so that we can mass deploy it and we can use it in the rural setups as well. And our devices would be useful for rural clinics, because they will have Internet of Things enabled devices which will transmit the data from those areas to tertiary care centers, and the specialist doctors from there can advise the patients regarding, after seeing their lung function, they can advise medicines as well. So we are integrating telemedicine protocols also into our devices. So I think that way, if we can make it available to the resource poor setting, it will lead to a good impactful innovation.
Participants, if you have any other questions, please ask. I think there is no more question. Thank you, Dr. Manas, for having a time for us from your busy schedule. Your session was so inspiring, and I think it is enlightened a lot of minds from the people, participants, and those are thinking for the innovation, and they are thinking in the startup area, and they must be inspire you from your journey. And congratulations for the Spyro Plus, and I wish the great success to that equipment, and a lot of people may be using in the India also. Thank you very much.
Thank you. Thank you. It was very nice interacting with you. Thank you for
and I request you visit our university. We are running the BTech biomedical and biotechnology students, and you take a session on that so that they get inspired and they can create their startups too.
Sure. Thank you.
So thank you, sir, for your wonderful session. Your insight into the advanced technology and their application in the healthcare have greatly enriched our understanding and the inspired to all. Thank you, sir, once again. So participants are requested to fill the feedback form before the 8:30 p.m. and we will meet in the next session with the tomorrow, the day 5. Thank you, all of you.
Thank you, sir.
Thank you, Dr. Manas.